Okay, welcome everybody. We're in a new venue this year. There's a whole bunch of activities going on today, and we couldn't use the Senate chamber, so we had the morning session here this morning, as we always do, but we're here this afternoon. So it's a bit more cavernous than the Senate chamber, but it's great to see everybody here and a good turnout. So everybody, I think, knows about Gairdner, so I'll keep it brief, just say a few words. Gairdner is who is sponsoring this for, I believe it's the 12th or 13th year at York now, a high school program in the morning and the York Gairdner lecture in the afternoon. So Gairdner is involved in the recognition of excellence in science, sponsor the biomedical, the most prestigious Canadian biomedical research awards, amongst the most prestigious in the world. So it's a pleasure to welcome everybody, and thanks for coming. The awards, Gairdner 60th anniversary this year, and it's York 60th anniversary this year as well. And I'll put a list of sponsors up shortly, but the awards are supported very much by a recent significant endowment from the Government of Canada. There are 3 Gairdner Awards, the Canada Gairdner International Award, and our speaker this afternoon is a 2019 laureate of the International Award. The Canada Gairdner Whiteman Award, and now entering its 9th year, I believe, the John Dirks Canada Gairdner Global Health Award. And so there are the 3 major awards. The Gairdner program that York is part of is unique to many awards in that this is part of what we call the national program, where laureates and former laureates speak across the country from Vancouver to St. John's and speak to, at most venues, not all, but at most, high school students to try and convey the excitement of science, biomedical science, research, professional, and just enthuse students about that. And so we did that this morning, and this afternoon we have the York Gairdner lecture, which is to faculty and trainees and is a research lecture. So the events are a true Trans-Canada celebration of scientific excellence. There's a series of symposia in Toronto as well as across the country. So this morning there was a global health symposium, and there was also a symposium on genome stability. And tomorrow, all of the laureates speak from 9 to 1, I believe it is, downtown. For those of you who are able to, it's a great event. For those who can't, it is videocast live, and on the Gairdner website, just look under events and you can get the URL for that. Tomorrow, as we're changing venues, the McLeod Auditorium at the U of T Medical School is not available this year, so the laureate lectures will be at the Mars Auditorium in the basement of Mars, 101 College Street, for those of you who may wish to attend. So the high school sessions are not really to teach science per se, but for our speakers to tell them a little bit about themselves, their journey in science, how they got to where they are, and the passion of doing science and what it takes to do science at this level. Again, everybody here, I'm sure, is aware of the excellent predictors of Gairdners for Nobel Prizes, and that reflects on the peer review, the two-stage peer review of the Gairdner programs. And about 30% of Gairdner Award winners have gone on to receive Nobel Prizes. And just about 3 weeks ago, I guess, the 2019 Nobel Prizes in Physiology or Medicine were awarded for work in oxygen sensing, and the 3 award winners were the same 3 who received the Canada Gairdner International Award in 2010. So, as usual, we beat Nobel by 9 years. That's not the goal of the Gairdners, but it's nice to know that our peer review process is exceptional and predictive and recognizing excellence. So I'm not sure if we could have the other slide up, the sponsors, but no, it was the other sponsor slide, but it's okay. Government of Canada, CIHR, Burroughs Wellcome, the Globe and Mail as the media sponsor, and York University are all sponsors, and we couldn't do these programs without them. And so just like to welcome everybody, thank our sponsors, and provide a brief introduction of our speaker today, who is Dr. Ron Vale, 2019, one of 5 2019 Canada Gairdner International Award recipients. He's a professor at University of California, San Francisco, and has recently been named as executive director of the Genelia Research Campus of the Howard Hughes Institute, one of the largest private funding organizations for biomedical research in the U.S., and he will take this position up early in 2020. He received his PhD in neuroscience from Stanford. Apart from the Canada Gairdner International Awards, he's received many other awards, including the Lasker Award in Basic Medical Research and the Shaw Prize in Life Sciences, and many more. In 1984, after studying nerve cells of the squid, he and colleagues discovered a new kind of motor protein named Kinesin. Over 15 years, Dr. Vale's lab worked on and deciphered how this motor walks along cellular tracts and made several breakthroughs with a different motor protein, dynein. So his discovery of kinesin and molecular motors transformed the field of cell biology, placing a spotlight on the study of motor proteins. His research has illuminated fundamental principles that underlie biological motility, an essential characteristic of all living organisms. The discovery of kinesin led to new tools for studying protein machines more broadly, sparked studies that connected motor proteins to many other cellular processes, and contributed to the realization that defects in these motor proteins underlie various diseases of the nervous system, heart, and other organ systems. And today he's going to talk to us about molecular motors from kinesin to dynein. I hope I've got that right, but it'll be on his first slide. So welcome to York, Ron, and thank you for being our guest today.
Okay, thank you for that very nice introduction. And of course, it's a great honor to receive the Gairdner Award in its 60th year. It's good because I'm also in my 60th year, so I guess we were born at the same time. And I think one of the really great things that the Gairdner Foundation does, which I think should be a model of all prize-giving organizations, is this great outreach program that occurs throughout Canada, because these awards should be really a celebration of science overall and an opportunity to communicate science very broadly, including to young people. So it's been a fun day talking to high school students this morning in this auditorium, and then had a nice time talking to some of the faculty and students, some of whom I recognize in the audience, and now to all of you today. So yes, like to talk to you about molecular motor proteins. Our lab actually works on other things now too. We work on the immune system, we work on RNA biology, but today I'll focus on this work that is the center point of the Gairdner Award. So as Ron also mentioned, why this went, motility really is a fundamental property of life. You know, even probably one of the first things that even nursery school kids learn about biology is that they see living organisms move. And of course, some of that is the way muscle works, the way animals can propel themselves through the world. But if you look under a microscope, you can also see a fantastic number of ciliated organisms moving by cilia and flagella. So it's a rich world of organismal movement. But also, if you look inside of cells, as I'll tell you about, and this is inside of a squid giant axon, you can see that it's packed with different kinds of moving membrane organelles. But this is true pretty much of any cell, any eukaryotic cell that you look at under a microscope. You look at the interior, everything's in motion inside. And of course, there are many other kinds of movement as well, including cell division. And here's the process of mitosis, the chromosomes in green. It was fun talking to Art today about other kinds of mitosis over lunch. But this is a very classical form where you can see the chromosomes separating from one another. And all these types of motility, in a way, have analogies, I think respectable analogies to how we undergo transport needs in a city, for example. So goods are made in one place of the city that have to be transported to other places. Of course, there's a lot of regulation of how that traffic moves. There are cars carrying various cargoes. And inside of cells, there are also forms of roadways, either actin filaments or microtubules. And along these roadways or filaments, there's material transport. These are membrane vesicles, and there are specialized machines called molecular motors that move things along these tracks. And all that motion that I showed you in the first slide is really based upon these kind of molecular machines. So as we've learned over the years, these motors are also part of large gene families. These show the three major classes of molecular motor proteins. One are the myosins, which are most known for their roles in muscle movement. But there are numerous different kinds of myosins in a human genome that are executing other kinds of transport along actin. Along microtubules, there are two major classes of motors, kinesins and dyneins. Again, in the human genome, there are many of these. And so what we've learned is that evolution has created not just one type of motor, but many different kinds that are specialized for different kinds of motility functions. So what I'd like to do in this lecture today, well, actually before that, I just put out a general word of, why is it interesting to study these? And so, first of all, I think what we've encountered now in biology is that, as Ron mentioned again, motion is so central to so many biological processes. And in a way, many people have joined the motor protein field because they were immunologists studying a certain process, and then they figured out that a motor protein plays an important role in their function of their cells, or developmental biologists encounter motility as a special way of moving RNAs inside of embryos, in neurobiology, et cetera. So it really touches upon all major fields of biology. A second important reason, I think, is that these molecular motors have really served as paradigms for understanding protein machines more generally. And we'll come to that. But I think this is very important for biology overall. I know there's a big boom right now in omics, in genomics and proteomics and all kinds of omics. But at the end of the day, we need to know more. We need to know not just whether genes are on or off or how much RNA is being transcribed, but we need to understand what is being produced by these genes. What are the proteins that are being encoded? How do they work? How are they regulated? And I think to really deeply understand biology, we're not going to be able to bypass that step. We need to understand protein machines in great detail, and I think motor proteins have been kind of, many of the tools that have been developed for motor proteins have proved applicable for other protein machines as well. And of course, I will say, in addition to the fact that I think they are fundamentally interesting, I think a lot of people are interested in motility, but it is of medical importance too. There are many diseases of the nervous system, of muscle, et cetera, that have defects in molecular motors or proteins related to molecular motor function. And in addition, I think there are opportunities to manipulate these molecular motors in ways that can treat disease. And one example of this is actually from a company that I ended up co-founding with James Sabry, Jim Spudich, and Larry Goldstein, called Kinetics. Actually, my very first graduate student was the first employee of that company, Fady Malik, and he has led this amazing drug discovery program, which I'll summarize more briefly, but it was about finding drugs, small molecules that actually activate cardiac myosin, so that's the motor that makes your heart contract, and actually activate that molecular motor. The drug binds to the motor directly, activates it, and actually increases the performance of that motor, so it produces more force-producing cycles per unit time. And that is useful for many patients that suffer from cardiac failure or heart failure. And that's a disease that affects many, many people across the world, where the heart muscle is no longer contracting as well as it should, and as a result, there's impaired heart function, blood flow, et cetera. And so this drug is now being used in a phase 3 clinical trial. So now 8,000 patients all over the world are enrolled in studying this drug. And on the left shows an echocardiogram where you can see the mitral valve there of a patient with heart failure, and it's kind of fluttering. And that's because the heart isn't contracting strongly enough to project adequate blood flow across the chambers of the heart. And after several hours on this drug, you can now look at the same patient with the echocardiogram, and now you can see that valve actually kind of popping open, and that's due to the increased contractility of the heart. So what this phase 3 study is about, we know the drug works, we know they know, I should say, exactly how it works, where it binds, the detailed biophysical mechanism of this drug. And now, the goal is to see if this reduces mortality and hospitalization in these 8,000 patients. But none of this would have been possible without actually understanding the details of how molecular motors work, their biochemistry, their biophysics, their structure. All of that was necessary to even start a drug discovery program like this. So what I'd like to do in today's talk is really go through a little bit, summarize very briefly some past history about how kinesin and dynein actually work. I'll then describe a more recent study, again, relatively short form, about how motors are regulated and how they attach to cargo. And if I have a few minutes at the end, I did talk to some of the faculty, but I'll just overview of some of the science communication efforts that I'm involved in as well. Okay, so I'll start with the era of Kinesin. This began with me as a relatively much younger man than I am now, in the early 20s, being a graduate student, whose advisor was Eric Schutter, and I'll describe how working in his lab got me initially thinking about this problem. But then I ended up traveling to the East Coast to the Marine Biological Laboratory at Woods Hole, Massachusetts, to work with Mike Sheets, Tom Rees, Bruce Schnapp on this project that led to the discovery of Kinesin. But the problem that got me into this from Eric Schutter's lab was as follows. Actually, I was studying a hormone called nerve growth factor, which Eric's lab was working on, and nerve growth factor interacts with the nerve terminal. It interacts with receptors at the very end of a nerve cell. And so we're interested in how the receptor works and how the signaling works. But there was also the realization that the interaction, that a signal had to be transmitted from the nerve terminal back to the cell body to control gene transcription. And of course, there was also the realization that not only do signals have to go from nerve terminal to cell body, but materials have to also be transported from cell body to the nerve terminal, because most protein synthesis and lipid synthesis occurs in the cell body. So there had to be two-way trafficking of materials across the nerve cell. And this is particularly interesting because the axon of a nerve cell can be very long. And in this case of a motor neuron in your leg, it can be 1 meter in length. So on a molecular scale, these distances, like 1 meter, are an extraordinarily long distance. So it was known that there was a transport system. People have measured it. They injected radioactivity, for example, in the cell body and can watch it move through the axon. But the mechanism of how that transport worked, really when we started in that photo a long time ago, was not really understood. So to approach this, we needed a good system, and that system, and also the reason for going to Woods Hole, was the squid and its giant axon. So the squid has also played a very important role in neurobiology history. It's also where Hodgkin and Huxley determined how electrical conductance works in an axon. And the reason why it's been a model of choice is that the axon of the squid, there's one axon that's 50 to 100 times larger in diameter than a human axon. And that means you can dissect it out and study it, and even in our case, it was valuable for doing biochemistry. So I think that the general approach to this problem was to take an initially complex system and then try to simplify it. So the complex system, of course, is the whole axon. And what you're seeing here is a video microscopy, which was actually pioneered by Robert Allen and Ray Lassick and Scott Brady. But you can see the inside of a squid giant axon here and see lots of things moving in it. But as you know, a cell's very complicated. There's lots of things in there. We wanted to know what the basic machinery was. And the goal was to basically take this system apart through reconstitution biochemistry in a test tube environment, which ultimately led to getting the transport system to work in a test tube, and later included then even replacing the natural cargo, which are the membrane vesicles, with artificial cargo In this case, what you're watching on the right, although it looks like the native system, what you're seeing here are plastic beads, artificial man-made beads of about less than a micron, moving along what we later discovered were microtubules using some biological machinery. So now we can get the system working outside of a cell. We could start replacing the cargo, and we're kind of on our way to defining what the machinery is. And then a next step was actually to figure out what the motor that was driving this kind of motility was. And that was done by like classical biochemistry, fractionation, and so forth. And this just shows a late stage gel of a protein that is correlates with movement that we could stimulate from those fractions in this test tube environment. And that protein was completely new, and we ended up naming it Kinesin. I told the high school students this morning that my mother Evelyn Vale, played an important role in naming it, because she actually consulted with a Greek friend of hers to come up with the name Kine, which means movement. But actually, looking back on it, it was also kind of interesting, because this was one of the, I didn't think about it at the time, but biochemistry was largely done with radioactivity or spectrophotometers or things like this. And this was actually using a microscope as a readout for biochemistry. And that's something that I've ended up kind of using throughout the rest of my career as a strategy. So, and even you'll see here. So the next question is, we reconstituted the movement, we discovered the motor, how does it work? And I also told the students, I believe, that 1985, 84, when this work was going on, it seemed really unimaginable to be able to answer that question, because we can't even see the motor in this image here. These motors are, 100 millionth of a meter in size. And, how do you actually figure out the real details of how a machine like that works to produce this motion? But like many things, there was a game plan, but there was also new technology that developed along the way. Like reconstitution, that we started with, but continued to be a valuable strategy. The motor protein field in some ways helped to launch the single molecule movement of biochemistry. In fact, the very first single molecule assay was patch clamping in ion channels, then a big hiatus, and actually, I think the next paper in that field was something that our lab was involved with in developing a single molecule assay for Kinesin. So now that's very common, but I think molecular motors and also myosin and so forth helped to kind of propel that single molecule field forward. And then, of course, we also needed to know in detail what these things look like. And again, X-ray crystallography, cryo-EM, all became kind of much easier than they used to be. So as an example of this, I showed you that bead assay, but we then could, with Toshio Yanagita, actually when I was on sabbatical, replace the bead with a fluorescent dye. So now there's no cargo, there's just the labeled motor, but we can follow these using kind of a new microscopy at the time, which is not so pretty ordinary right now, called total internal reflection fluorescence, and very sensitive cameras could see these single protein machines walking along the track. There were techniques developed by Paul Selvin and others where we could track those fluorescent dyes with great precision. We can see the motor step. So we can now measure the dynamics of that. That still didn't answer the question of how the machine really worked to produce these individual steps. But we knew that the steps had to be choreographed in some ways, that these machines are ATPases, so they're enzymes. They're hydrolyzing ATP, they're releasing phosphate, and then releasing the ADP, and then cycling back by grabbing hold of another ATP. And we also knew that during each of these cycles, the motor would take one step. And then the question is, like, how did it do that? So for that, we then needed to go to really know what the motor looked like. We did that with Robert Flederik and colleagues to get a crystal structure. Of course, the initial crystal structure was just in one state. So the field overall, just like watching snapshots of a horse, you have to kind of understand the different states of the horse as it gallops. That was kind of the goal we were trying to get to with motor proteins as well. But the other thing we learned in our journey along the way with the protein structure, which helped us enormously, is learning also by comparing Kinesin to other kinds of machines. And we learned some kind of common themes that run through a whole variety of machines that previously people thought were unrelated. So for example, even the myosin field and the Kinesin field largely never talked to one another because they all thought that their motors were special and different. Of course, one moved along actin, the other one moved along microtubules. And what we discovered, although I won't show you the data for that, is that even though the sequence of Kinesin and myosin totally different, even though one moves along actin and the other one moves along microtubules, the structures were clearly similar to one another, particularly in the core of the enzyme. And in fact, they were more similar to one another than, Kinesin and myosin are more similar to one another than to any other protein in the genome. Not only that, but we also ended up discovering that the motor proteins, Kinesin and myosin, were also similar to G proteins. Now, G proteins people don't think of as like necessarily as a molecular motor, but in a way they are. And they use energy from nucleotide hydrolysis to convey information. So they convert energy into information flow in signaling pathways, for example, like RAS, or in the ribosome to make proteins. There are a whole variety of ways which G proteins are used. But what they both have in common are they take a triphosphate nucleotide and hydrolyze it to a diphosphate nucleotide. And they change their protein shape when they do that. And that change in protein shape is used in a strategic way by the protein either to communicate information or to produce mechanical work. And in fact, if you think about this evolutionarily, probably what happened in evolution is that there was a great invention, God knows when, but a very long time ago in early history of life on Earth, where some protein was developed that learned to be a switch based upon chemistry. As I mentioned, it could switch between states whether there's a triphosphate nucleotide or a diphosphate nucleotide in the pocket. That was a great invention, and it was used in a couple different ways. One is to make this family of G proteins, and in another branch, another protein branch emerged that was some kind of precursor of a motor. We have good evidence that it historically existed, although we don't have an idea whether there actually exists a modern version of that motor protein precursor. But we know that Kinesin and myosin clearly, Kinesin did not evolve to myosin or myosin to Kinesin. They both evolved from a common ancestor. And that proved to be a pretty nice invention. And then after that, those motor kinesin motors and myosin motors proliferated to create many different genes, as I told you about earlier, to make many different kinds of kinesins, many different kinds of myosins that work in the cell. So this is what we think is the evolutionary history. But from the evolution point of view, I'll give you a brief tour of how we think all these proteins work, how motors work. So the first thing I said is you have to be able to tell the difference whether there's a nucleotide triphosphate versus a diphosphate. So these machines have to tell the difference between the presence or absence of a single phosphate group. So that's how they work. How does that occur? Well, this is kind of the core scaffold of both myosin and kinesin. They pretty much look the same if you project them at that level. And G proteins. And there are two loops called switch 1 and switch 2 in the field. And these are loops that are sensors. When there's 3 phosphates, ATP or GTP, those loops move in and they grab hold of the terminal gamma phosphate. When the gamma phosphate is cleaved enzymatically, the phosphate comes out, the loops move back. So the movement of these loops in and out, they're small movements, but those are sensors of the nucleotide state of the enzyme. Next, that information, so what I told you about is now projected in the whole protein structure. That blue core is what I showed you in the previous diagram. That's the part that's the sensor, but that sensor now has to be relayed to other parts of the protein. And that's done in both cases by a very similar, it's very sensitive, by a very similar helix called a relay helix. And you can see it's in the same position in both of them. And that helix is basically a rigid rod that slides back and forth. And it's attached to one of the loops. So when one of the switch loops moves in, the helix slides along with it. When that loop moves out, the helix slides out. And because the helix is a rigid rod, it's a great way of communicating information across long distances, relatively long distances across the protein. Kinesin and myosin, G proteins, all use this same strategy. Now, the one difference that most G proteins don't have and motors have is they have a mechanical element, something that can take that nucleotide information and now create some kind of mechanical movement out of it. And that is these mechanical elements that you see here. In fact, the myosin field knew a lot about that mechanical element. We didn't know where the mechanical element was in kinesin when we got the structure. Well, what do? We just looked what is in the same place at kinesin relative to what is the known lever arm in myosin. And that's how we made a guess of where kinesin's mechanical element is, which is this little yellow element that you see there on the left. It's basically right at the end of that helix, and you can see in myosin that yellow element also is right at the end of that helix. So basically by using evolution in a smart way, we kind of bypassed years of research to make an educated guess. And this is, first of all, to say, here's how the myosin mechanical element works. Myosin binds the actin, the phosphate comes out, the nucleotide sensor kicks into motion, and that big yellow element swings like a big rigid ore, about 10 nanometers, and then it dissociates and recocks. And it's literally trillions of these binding, stroke, release events that make your muscle contract, all by each motor producing a small movement, but then it's amplified over like billions of these machines in your muscle. Okay, so we then study this mechanical element in kinesin in great detail. This just summarizes a huge amount of work in an animation. But this little element exists in either a red state where it's detached pointing backward, but you can see when ATP comes in here, this element now wants to zipper up along the core of the motor domain. And when it zippers up, it causes, because it's attached to the partner motor domain, the partner motor domain releases, and the zippering causes the partner motor domain to move forward to a forward binding site. So the two motor domains play kind of leapfrog action, or you could think of it almost like also like a judo expert. That judo expert grabs, partner, and with a flip of the arm can throw the, opponent forward, and that's kind of what the Kinesin does with its arm over and over and over again, and it walks in this very regular way. So, that was about 15 years. Seems like a long time, but as I said at the beginning, I thought I'd be 60 years old and I wouldn't have any idea how the motor works. So in that sense, it was fast. But also just to say, there are so many people involved in this work. I named some. I can't name them all, but there have been lots of amazing colleagues and graduate students and postdocs and collaborators who all participated in all of that research. And of course, all these Gairdner awards, they're given to a person, but they almost should be thought of as given to a field because a lot of people were involved in these work. So the other thing to say is that, so we had a good idea how Kinesin works, but there's another microtubule motor, which at that time, around 2003, was very little understood. It was discovered many years ago. It was called dynein. Kinesin moves in one direction from the spinal cord in the axon down to the nerve terminal, but dynein walks in the other direction along microtubule tracks. It also was originally discovered as the motor that powers the beating of cilia and flagella as well. So it's involved in very important motor, involved in many different things, and so we decided to start working our focus on dynein. And again, I would say we basically use the same playbook. It's a good playbook. It's still a good playbook, which is to try to reconstitute things in vitro and test tube where you can control things, develop single molecule assays to understand dynamics, and build up an understanding of atomic structure. Now, the one thing is you could say, well, you figure out Kinesin, so dynein is another microtubule motor. It's probably going to work the same way. Wrong. Amazingly, even though I told you Kinesin and myosin are related to one another, dynein, although a microtubule motor, comes from a completely different evolutionary origin from the G proteins and the Kinesin and dynein motor. It comes from another group, which you may have heard of, but are called AAA ATPases. Many of them are involved actually in protein unfolding. So dynein is kind of a weird uncle in this family in a way, because it's learned to use this enzymology to create a mechanical event. Now, the other reason why this study wasn't easy is also, and the reason why dynein was farther behind, is it's a behemoth motor. It's about 8 to 10-fold bigger than Kinesin, you can see on this same scale, and also much bigger than myosin. So there were a lot of challenges in expressing the protein and studying it and labeling it, all these things. But it's a fascinating motor, and I won't go through all the details here, but it has this ring, ATPase ring, and the main ATP site is way at this other end of the ring, but yet it has to communicate information all the way down this long stalk to this foot, and that's where the microtubule binding domain is. And the motor has to bind tightly, but when you walk, it also has to release and rebind and so forth. So the information has to be, of the enzymatic cycle, has to be communicated over this long distance of about 25 or 250 angstroms, so it's a long way. And there's a mechanical element in purple here, and that information has to coordinate the actions of that mechanical element. And I would just say we spent a long time, also we and other people in the field, the dynein motor takes all different kinds of walks. So the Kinesin was a very regular stepping motor, And dynein has kind of a very strange walking motion, but it's almost like a giant here on stilts. And we think the conformational change is involved in changing this yellow element. You'll see it flip to a bent red state right there. And when it does that, it throws basically this long stilt and leg forward along the track where it can rebind. There it is again. But unlike kinesin, where the two heads are coordinated, the two heads of dynein seem to be working completely independently. It also can do unusual things. Sometimes it goes forward, but it takes a lot. It also takes backward steps, which kinesin very rarely does. And it also can switch even the orientation of which head is on which side and move kind of laterally along the track as well. So anyway, I think now, again, also, these are many of the great postdocs in the lab who contributed to this. I think all of them are running their independent labs right now and doing fantastic work. So I'm kind of leaving the dynein field to them right now because they're doing such amazing work in this field. But one thing that we're interested in now in the lab is how these motors are regulated. So not just the biophysics of how it moves, but just like transport in a city, things move, but things move in kind of a regulated way. Actually, what you're seeing on the right is cell from the skin of a fish or amphibian. And amphibians and certain fish can change their skin color, and that's due to pigmented organelles that either are moving in or out by molecular motors. So they do that in a controlled way with nerve stimulation. But it just illustrates the notion that motility has to be controlled. So one thing we know about at least the transport of membrane vesicles is that there are switches that govern the recruitment of motor proteins to organelles. So I talked about G protein switches. RABs are G proteins that switch between GTP and GDP states on membrane organelles. And one of the things they do is to recruit motors. They do other things as well, but one of the things they do is recruit motors. And first what they do is if they're in a GTP state, they recruit a coil protein, which is kind of a linker protein. One end of it binds to the RAB protein, and the other end of it binds to another kind of monstrous regulator of dynein called dynactin. And when the adapter binds dynactin, it then can also bind dynein. And this is, in fact, the motile machine. It's not just dynein, it's this whole complex that moves membrane organelles. So Yuxiao Wang, a more recent story. He's actually just moved from the lab very recently to found a startup company, actually. But he was studying to see if he could find new dynein regulators in the RAB family. And what he did is just do a bioinformatics search to look for unusual RAB proteins. And he found a couple that were unusual because most RABs are just that red piece that you see on the right. But a couple of them were extraordinarily much bigger than all the other RABs. And he looked at the sequence and said, oh, wow, there's a long coil protein in that RAB. He knew dynein adapters were also coil proteins. So he said, okay, instead of having an adapter protein in a RAB, what if the RAB and the adapter fused together to create an integrated gene that was an integrated dynein adapter RAB protein? So this was the idea, is this an integrated dynein RAB? The other part of the protein also was very interesting because it's an EF hand, which in many cases is known to be calcium regulated. So here was a theory to test, and let's go to the playbook of reconstitution. So that RAB protein is called cracker2, a name that kind of came out of the literature. There's very little known about it. And he purified all these proteins and see what they did. So first thing is, by IP, yes, the cracker was able, that's the Rab protein, was able to pull down dynein dynactin, and it did so on the right column in a calcium-dependent way. And even more excitingly, if you mix the proteins together and fluorescently label the dynein, if you look at it with no calcium, there's no movement, the same test tube now adds calcium, and now you can see this, the dynein dynactin and the Rab protein moving along the microtubule tracks. So this in vitro experiment indicated that this theory that this could be a dynein adapter protein, a regulator of dynein, proved true, and the calcium regulation also proved true, and this is the only known calcium regulator of a microtubule motor, to our knowledge. There may well be more. He then looked to see if this calcium regulation is true in cells, and I'm kind of simplifying a long story, but if you fluorescently label this cracker protein, you find it on some membrane compartment inside of the cell. It's some endosomal compartment we still don't understand. But if you cause calcium to flow inside of the cell, either with an ionophore or actually through an endogenous signaling activation pathway, which releases internal stores of calcium, these endosomes now will move along microtubules to the cell center. So that's what you see here. This very tight dot there is all these membranes that have clustered right around the centrosome, where the minus ends of microtubules are. Okay, but then, you know, there's always a story at the very end of your study that you don't completely understand, and I think we've left this story with kind of a cliffhanger to something new that we haven't completely figured out, but I think is an exciting direction. When we looked at these cells by this technique called turf microscopy, we found that this new cracker Rab dynein adapter protein was not only in the intracellular membranes, it also got recruited to the plasma membrane. And this isn't a T cell, and it would only get recruited to the plasma membrane once the T cell receptor signaling pathway was regulated. So you activate the TCR, and what you find are these little spots of this dynein adapter that get recruited and form on the plasma membrane. These spots co-localize with dynein, and if we follow these spots with another kind of microscopy, which is spinning disk confocal microscopy, which gives you a view of the interior of the cell as well. The one on the left just shows the plasma membrane. But you can see some of these dots. You can see, I don't have a pointer here, I'm not sure this pointer works, but you can see just below the C over there, there's a green dot that appears. It forms in the actin-rich cortex, and then right now, boom, it moves along a microtubule track. And I'll just simplify to tell you what we think is happening, although we don't have definitive evidence for this model that I'm going to show you. But I think it's exciting. Those of you that know endocytosis know that actin is involved with endocytosis in many settings. But we are wondering if there may be some other kind of endocytosis where the mechanical force of pulling a nascent endosome off the plasma membrane is driven by dynein and microtubules. And that this dynein adapter that I told you about, this cracker, may be involved in this process. So the idea is that induced by the TCR signaling pathway, this cracker protein binds to the plasma membrane, and we don't understand what regulates that process. And it must bind to some specific types of cargo that are destined for endocytosis. We know one of the cargo that it's transporting inside the cell, but we don't know the complete spectra of what other kinds of molecules it's pulling in. But then it's recruiting then dynein, which we can see, but the model is that the dynein recruitment is actually getting this nascent endosome onto a microtubule track and pulling inside the cell and delivering that cargo and that endosome to the very cell center. Lots of mysteries here. Well, first of all, we don't know if this model is completely right. We don't know why T-cell signaling is inducing this. We don't know why microtubules are involved in this particular pathway. We don't know why that cargo and that endosome has to be delivered all the way to the very cell center. So I guess, you're all scientists. Like every story, there are new mysteries to solve. And, this is my current lab group. They're involved in lots of different projects, but as you know, like one of the real joys of this whole business is to be involved with young people who are just so much fun to interact with, and I think we have a lab that really has a lot of fun together, and that was a picture taken not that long ago, and I'll just whip through this very quickly, but I also spent a lot of time thinking about science communication. And also, how do we make these discoveries accessible to many people? I founded this project called iBiology. One of the goals of this project is to film lectures of world-leading scientists in a studio environment and make these talks freely available throughout the world. So if you don't know about it, you should check out this website. There's an amazing group of lectures that are on demand. Available to you anytime, and it really is like an amazing seminar series. I talked to some people about this project. This is a new free, I use the word textbook in quotes, but it's a resource for college biology to teach students what science really is, and that science is not about memorizing facts. It's about problem solving, it's about detective work, it's really about, the really interesting aspects of how people figure out how things work in living systems and how wonderful living systems are. So have a look. This just launched a couple of weeks ago. It's www.explorebiology.org. It would be great if York University starts, using this to teach some of the college students here, but I think you'll enjoy some of these stories yourself. You'll see that Nobel Prize winners and a lot of really great scientists have contributed to the content, and I now have a grant, so we'll try to develop more content over the next 5 years. So it's a space to watch, and we, through iBiology and two very talented filmmakers, Elliot Kirshner is now working as the executive producer of iBiology, made a film on genome editing that just appeared in Canada, was in Toronto, I think, last week. It will be on Netflix and other venues like that, but this is an effort to try to credibly deliver scientific discovery and information, in this case about CRISPR, to the general public. So this is an hour-and-a-half documentary. I think it's really good, and I would encourage you to look for it later. It should be coming out more visibly next year. It's been through film festivals, including Hot Docs, which is here in Toronto. And I also founded another organization called ASAP Bio, which is to try to make the lives of all the graduate students here and postdocs hopefully much better in a few years, to improve the scientific publication system. Again, this is a grassroots organization, recognizing that we as scientists have to take control a little bit and create a scientific publication system that's really working in the best interest of science, best interest of the public, and that's what this organization's about. It's now a 501c3. We've advanced preprints, but we're also started a new peer review process, which is called reviewcommons.org, which is being partnered with EMBO and PLOS and eLife and several other journals. So it's launching in, I believe, December. It's actually launching at an ASCB meeting for those of you that are going there, but you can get a sneak peek of it on the website of what's coming. But anyway, I think it's also very exciting, experiment and scientific publication. And here's where I'm going to next. Talk about more adventures, Genelia Research Campus, as Ron mentioned. And just to tell all of you, it is an amazing place. I mean, that's why I'm going there as executive director. And it's an awesome place to be a postdoc or apply for a group leader position. I'm happy to talk about that as well. So again, thank you for all of your time. I probably went over time. I wrote this essay on how much I love science, and I'm happy to talk to you about that as well. So thank you very much.
Okay, we have time for a few questions. Let me just say something briefly about one of your last remarks. I actually saw Human Nature at Hot Docs, I guess 2 weeks ago, and it's very, very well done. It's interesting. The science is interesting, and the ethics and the social science. It's worth seeing if it comes to regular theaters. I think it showed at Hot Docs about 3 times, and I presume it'll be available in other ways soon. But it's worth the view if you have a chance. So some questions, if we can see.
Yeah, I can see pretty well. I'm adapted. Yeah, there's someone.
Rulan, thank you very much. I wanted to ask about the clinical trial.
Oh, yeah.
You made a comment earlier about sort of use in neurodegenerative diseases and other types of diseases. For the motor proteins, though, don't you need to diagnose very early in the disease before you can actually make an impact with the drug to stimulate the motor proteins? My assumption would be if you're too late stage, can't, like the cells, function at that point? Or do you really think that there's regenerative capabilities within the neurons?
Well, okay, so that clinical trial that I showed you there, that was on heart failure. So right now there are no motor protein drugs in, neurodegenerative diseases. And I think the question that you raise is a very good question, not just for motor proteins, but all kinds of other strategies for thinking about neurodegenerative disease, and that is how reversible is it? I think, in the case of, for example, there are neuromuscular diseases, and like muscular dystrophies, and muscles have a lot of regenerative potential. And I think there the potential for reversal is great. I think it's, I think this is kind of the central question in the whole neurodegenerative field. And of course, really there's very little, if any, and I would say truly effective drugs for, diseases like ALS or Marie-Charcot-Tooth or all these kind of we're just in unknown territory. There's several challenges because in neurodegenerative diseases, there's probably so many things that are wrong. One thing triggers another thing that triggers another thing. So even like knowing what is the piece of biology that has the best chance of reversal is not known. So obviously there are various companies thinking about that, have different ideas, but I don't know. I actually don't know the answer to that question, and I think that, of course, that's why we do science and we do clinical medicine, and maybe we'll be surprised, and maybe if some of the fundamental biology of getting nerve transport to work much more efficiently, and once clogged pipelines are unclogged, maybe things can be restored. But it's. But I think we'll probably know the answer to this in the next 10 years because there's so many companies focused on this question now that we're going to know a lot more about that. Yeah.
So what do you think nature evolved a very simple, say, Kinesin motor, good cargo one way, but a very complex, bulky, dining protein, which you would think the opposite way, or the opposite direction? If you look at sort of an evolutionary basis, where these proteins may have started,
Yeah, I think dynein was evolved mainly to vex biochemists. I think that's probably my best answer to that. Well, seriously, it's a very interesting question. It's kind of hard to second guess why evolution works the way it does. I will start by saying, though, that maybe higher plants were the smartest creatures on this planet because they took one look at dynein and said, wow, this is too complicated, and got rid of it. So in fact, even though in the earliest eukaryotes that people find, Giardia or other things like that, all those organisms have dynein. And yet, interestingly, higher plants got rid of all the dyneins, all the dynactins, and they replaced the main. There's kind of one main dynein motor that drives traffic in the other direction along microtubules. They replaced it by expanding another family of kinesins that move in the same direction as dynein. So there is a group of kinesins that can move in the dynein direction. So you don't need a dynein to move in that direction. Even yeast have developed a kinesin that moves in the dynein direction. Humans have 3 of these -n-directed kinesins, and plants have about 25 of them. So their solution to getting rid of dynein is to expand the kinesin family, specifically in the motor subgroup that works in the dynein direction. So that just proves life without dynein is possible. Plants have developed it. And why dynein emerged to begin with is not clear. There may be some very significant advantages of dynein that we don't understand, not so much for transport inside of cells, but maybe for cilia and flagellar function. The beating of cilia and flagella is a very complicated process. We still really do not understand how that works in terms of generating these waveform patterns. But it's possible that also dynein was part of the evolution of these motile cilia, which probably occurred also very early in eukaryotic life as well. But otherwise, it's a question don't have a perfect answer to that why question for evolution of this very complicated machine. And even its friend, dynactin, dynein is about 2 million Daltons. Its regulator is about 1.2 million Daltons. And it's an incredibly Baroque thing, too, the way it looks. And why that thing is so complicated and Baroque in structure, also not entirely clear, at least to me. But, sometimes biology does that.
Okay, any last questions?
I'd like to follow up the previous question. Are those kinesins that what was the mice and that involving mitosis, bringing the chromosome to the central?
Yeah, well, there are 2 ways in which the -n motors are used in mitosis. One is dynein, and also one of the family of -n kinesin motors is involved in the shape of the spindle. So the spindle has often this football shape, and the -n motors kind of move to the pole and kind of hold the pole together. So they're involved in the shape of the spindle. The dynein motors are also involved in a checkpoint mechanism for the metaphase-anaphase transition, where it's kind of a complicated process, but they are transporting proteins off of kinetochores onto microtubules as part of a checkpoint system for mitosis. And the third part is like the physical movement of the chromosomes. Well, Art has done some very nice work on that, if he's here. But, huh?
He just left.
Oh, he just left. Okay. Well, I think work from his lab many years ago also suggested that depolymerization of the track is actually part of the mechanism for the shortening of the track and the movement of kinetochores, movement of chromosomes to the pole. So some of that motion may not be driven by motors, but involves actually the depolymerization of the underlying roadways.
Okay, question back there, and then maybe we'll.
I was just wondering, with the new form of microtubule-dependent endocytosis with the cracker, could it have any medical applications in immunology, and what kind of cargo we want T cells to pick up, like, in the future?
Yeah. Well, you know, this is really raw basic discovery at its rawest nature, in the sense that that's why we do basic research, is we don't know where it's going to end up, or we don't know what's going to be medically important. I mean, right now, we don't know what the cargo is or what the purpose of it. Of everything I showed you, we have no idea what the purpose is. So, you know, right now, we can't develop anything, a medical approach to it, because we don't understand the fundamental biology, and that's always the place to start. I mean, once we understand that, there may be an aha moment. Oh, I actually know what this is for. I can manipulate it, and there is a way to manipulate some readout or response of the immune system. So we're not there yet, but that's why we're doing what we're doing. Did you have a quick question next to you, and then we can close?
Yeah, this is about what we were discussing earlier. I had a question about the fact that dynin has a sort of more variability in the way that it walks towards the minus end, and I was wondering if that could have offered an evolutionary advantage compared to the fact that myosin walks in a more regular fashion. So
or Kinesin. Yeah. So, I mean, one thought, and there is some data from other laboratories on this, that since these motors have to move in opposite directions, there may be an advantage if you have this small, regular motor. It just kind of plows along one track along the microtubule. It can't do anything else. Dynin, on the other hand, has these long legs. They actually stand up even above the motor domains of Kinesin. And the notion is it may be able to get around obstacles better than Kinesin. So if Kinesin and Dynin are heading on a collision to one another, Kinesin's just going to keep going, but Dynin can, kind of wiggle around to other regions of the microtubule and navigate around that track. So that has been shown in vitro and in other settings. Now, whether that is really true and happening in the neuron and is a reason for the different structure, that we don't know, but at least that makes sense as a possible reason why you would want two different motors with different stepping strategies.
Okay, so the day is yet young for those of us involved with Gairdner events, so there are a number of other things that Ron is involved with today, so I think we'll call it a day. Thank him again for this, and congratulate him again on the Gairdner Award, to add to many awards. And just to thank him for spending so much of his day at York with the high school students, with talking about his communication efforts, and hopefully York may be able to get involved with some of that and beta test things, if that's what you call it these days. Anyway, thank you all for coming, and next year, same time, maybe not same place. We'll see if we're back in the Senate chamber or not. But anyway, thank you all for coming, and another successful Gairdner Day at York.